All right, welcome everyone to a very special MStranslate broadcast. Hopefully you all know me. My name is Brett Drummond. I am one of the co-founders of MStranslate. And we do apologize for the slightly short notice about this live broadcast, but this is based on the fact that we were trying to secure, as we've posted, a very special guest for today, which we've managed to do. And so we are just going to wait a little bit of time and give people a chance to find the broadcast, come on board. So as you come on, please do say hi. Let us know who's watching. Let us know where you're watching from. And once we've given people a few minutes to come on and said hi, and we know that we've got a few people here, I will introduce our very special guest. So welcome, welcome. I can see that we've got a number of people linking in. Hey Charlie, welcome from Boston. Patricia, welcome from Sydney. Feel free to also take guesses about who you think our special guest is going to be. It's not me, obviously. Unfortunately, I'm not that special on MStranslate anymore. Hi, Deanna, welcome. Thank you for joining us. Also, if this is a time, we don't usually do live broadcasts at this time. If this is a time that suits you, a weekend lunchtime in Australia anyway, different parts of the world, different times. If this is a good time, let us know that as well, and we'll try and do these broadcasts more often at this time. But I can see we've got quite a few people coming on, which is great. So welcome everyone. Belinda, welcome from Geelong. Debbie, welcome from Geelong. Do we have some very happy people watching us from Geelong today, based on the results of yesterday's grand final? Not that everyone from Geelong is a cat supporter, but potentially you are. So we are going to be talking about a few different things today with our special guest, but as always, there's going to be an opportunity for you to ask us questions. So we're going to share some news, but whenever you're interested in asking questions, and today's really just going to be, we don't have a specific topic that we're going to be taking questions on. Anything about MS research will be okay. And so if we are ready, I might get around to introducing our special guest. I'm just waiting to get a thumbs up that he's ready to come and talk. So for all of those in attendance, without further ado, I'm going to introduce our special guest. No, that's who's now just going to try and mess with the broadcast. But he is someone who is very well known to MStranslate audiences, and I'm excited to say that he is here in Australia. You will all recognise this face, but Dr. Travis Stiles joining me today from Melbourne. So if you are unaware of who Travis is, he will talk a little bit about his stuff as well as we go through. But Travis is the CEO of Novoron Bioscience, which is a biotech company based in San Diego, California. Only a short 15-hour flight away, which Travis is looking forward to doing again on his way home. But Travis's company is involved in a number of different activities, but you will know him through conversations we've had before on MStranslate about their work in multiple sclerosis, looking into some novel remyelination therapies. Travis, welcome to Australia, although you've been here for a little while now. Why don't we start? We'll start with the easy questions. What have you liked about Australia? You've got to see a number of different animals. Saw kangaroos yesterday. How's your experience been finally making it to Australia after we've attempted to get you here a number of times?
Excuse me. It's been great. We've had some really cool meetings, and the beginning of the trip was a little bit rocky because I had originally planned to have this whole week to have meetings, and then I realized that you guys have a holiday for your AFL final, and then the Queen passed away, and so I had to condense a week's worth of meetings down into 3 days. So I ended up doing 4 Australian cities in 4 days, and that was not something I will probably do again. But I had a great time. We visited Sydney, Hobart, Adelaide, and now Melbourne. Had some great meetings, and I think that a lot of cool stuff's going to come out of it. I would say non-science, non-Novoron stuff. I love that you can. This is a dumb thing, but I love that like on a lot of mass transit, you can just tap your credit card. You don't have to buy a ticket or mess with things. I like that you guys have good mass transit. Um, I definitely enjoyed getting to see some kangaroos, and not at the zoo. We got to just drive out to the airport, and what was that place? Like some wildlife reserve?
Yeah, just a sort of wildlife park. Well, not even really a
That's like
wildlife park,
that's like
just a
just
park that the
outside
kangaroos
the
hang out
city.
in.
Yeah.
Yeah.
So that was cool. Got to go to the zoo. Got to go to AFLW footy game. That was fun. Now a Carlton supporter. Brett got me. I should have worn that on here. I should have worn my jumper.
It's okay.
Yeah, no.
You can, because we might alienate people if you put it on.
Oh, that's true. People might get mad.
Yeah.
But no, it's been a great trip. I just wish it wasn't so far away. It's a very long flight. It's a very long flight.
It is a very long flight, and we're both, I guess we can use this opportunity. We're going to delve into why you're here in a bit more detail in a second, but we can also sort of announce now, although we've touched on it a little bit before in other things that we've done, that Travis and I will both be doing long flights again in a few weeks' time to head to Amsterdam for the 2022 ECTRIMS meeting. And so we will be at the end of each day doing our video summaries, so you'll be able to hear all about what's going on at the biggest multiple sclerosis research meeting in the world that thankfully we're going to be able to attend in person again this year.
Yeah, we did this 3 years ago. We still have videos out there. That was a lot of fun. I can't believe it's been 3 years since we've been able to do another one.
Well, and I think the good thing about having both of us there, and the thing that we're excited about, is that one, you get double the opinions, but it also means that we're often in different sessions and hearing different talks, and so you get a much broader experience of what we're hearing at the conference as well. So that's one thing. We'll obviously be sharing more about that in the coming weeks, and obviously once we're on site in Amsterdam.
We're also going to be bringing on another special guest that we're going to be coercing into doing some content, right?
And hopefully more than just one as we're there. We're going to be having a number of conversations with people and trying to rope as many different people into interviews. But
I was gonna make him do stuff. Like I was gonna make him do this.
Right. He might. Well, if you can make him do that, yes. Travis has a research colleague that will be joining him there as well. Do you want to talk about?
Yeah, just in case people want to look into him. So it's Dr. Alban Gaultier from the University of Virginia Brain Immunology and Glia Center, where they famously kind of pioneered the discovery of the brain immune system. And a lot of his work is based. He's done a lot of the same work as I have on terms of remyelination. He's somebody that we've worked with closely on our remyelination program, and he's coming along with us this time. So hopefully we can have 3 sets of eyes instead of just 2 in the conference. Yeah.
Perfect. So let's shift back to current events. Now, obviously, we can't say everything about what's going on, but do you want to let people know a little bit around why you're in Australia, other than to see some kangaroos?
Yeah, so it's kind of exciting. So we are starting Novoron Australia.
Done. That's all we need to say. That's all we need to say.
So I could talk about this for a long time because we've been very excited for a while about the opportunity that exists in Australia, particularly with some of our MS research and especially with our connections to MStranslate and Brett's connections here in Victoria and beyond. We've long been thinking that there was an opportunity here for us to put some of our activities that have been San Diego centered for a while and bring them over here. And that's what this trip was mostly about, was setting up the subsidiary so that we could be a real Australian company and start figuring out what the future and expansion plans for Novoron could be. But in general, our main focus is going to be on centering a lot of our remyelination work here in Australia, with an eye towards expansion potentially into other indications as well. But yeah, I mean, and not to drone on too much about it, but Novoron has grown a lot since the last we did this in Ectrums in 2019, and we've now our drug development platform has been expanded, and we're now looking at being a much kind of more forward-looking kind of drug development company. And that's been really exciting for us. But it means that in order for us to maintain the traction in these important indications that we've already made a lot of progress in, I think that it makes a lot of sense to make sure that we have concerted, focused efforts. And I think that by being able to leverage a lot of the incentive programs, which we can talk about more if you're up for it, that Australia has to offer in centering a research team here, that we can give that the focus and the kind of emphasis it deserves, while taking advantage of some really great opportunities and incentive programs that Australia has to offer.
Yeah, yeah, definitely. So, I mean, it's exciting, I guess, for our local audience, especially, that we're going to have some remyelination research happening down under. There's obviously other remyelination research happening here, but to have Novoron and Travis's expertise shifting some of their research goals down under is something I've been working on him for a while to do. Keen to get it down here, but so exciting. George has asked if we're hiring.
We will be.
We will
I
be.
don't know what that looks like yet. We're still looking at where what the kind of team build out and what the headquarter kind of like search situation is going to look like. I think we're looking mostly at the Melbourne area, and potentially having some activities down in Hobart as well, interestingly. So we absolutely will be hiring in the near future. What the near future is, I'm not positive. Things don't ever move as fast as I would like them to, but there will definitely be Novoron employees in Australia over the next 6 months.
Yeah, I mean, I think it's important to say this is obviously at a very early phase. This first trip was really about doing some fact finding, having some meetings, starting to think about what that looks like, but with a commitment to start to move towards having some of that MS research here. So,
and I think I do think, I don't want to hog the program, but I do think
the program is about you, so you can definitely hog the program.
That's just weird and unfortunate for everybody. But I think that a lot of the learnings, a lot of the fact finding we did here, I think we learned a lot of really interesting stuff that the MStranslate audience could be interested in. I'm obviously very excited about our stuff and what we want to do, but it's not an easy thing for us to say that we're going to uproot some of our team and move them, or create activities, on the literal other side of the world. And I think that people would rightly be questioning, like, why would anyone do that? And I think that that's, I think that people that are from Australia in particular, but anybody that's interested in the research field and MS field, I think they there's a lot of things that Australia is doing that is really, really cool in terms of being able to foster innovation. And I think that there's also some gaps that I think that people in this country, if there probably could merit a little more talking about, so that people could advocate for maybe filling in some of those gaps, because I think Australia has an incredible amount of potential right now to be a leader in scientific innovation going forward, in a way that I don't think I necessarily appreciated before coming here.
Yeah. I mean, I think some of those nuances are probably for another discussion, considering we have limited time today. But I think, I mean, to summarize that there is part of our discussions over the past week have not only been about let's help Novoron get here so that we can do some of that work, but also as trying to help remodel the landscape a little bit, so that Novoron's not the only one that can do that, and that more life sciences companies and biotech companies working in the health space see this as a good option.
Yeah, look, I'm a big fan of rising tides lift all ships. And I believe, look, there's basically two ways to look at Australia. There's really good incentive programs where I could come here, do a very narrow, focused kind of set of activities, and I could benefit from that. And then when I'm done with those activities, be done and potentially move on, find another thing to do, or I could invest in something that has actual growth potential here. And I actually envision the growth potential option is the better option. It's just there are components of the kind of ecosystem, the scientific ecosystem in this country that could be improved upon that would make it easier not only for Novoron to envision a much more aggressive growth strategy in Australia, but also foster more innovation. Because one of the things you don't want to do as a small biotech company like ours is be the only one. Like there's a tremendous benefit of community, and you guys have a tremendous academic community, and there's a lot of efforts to create more of a industrial opportunity, spinning out technologies from the universities. But I think there's a lot of simple things that could be done that could make that system more robust and sustainable, instead of, I think a lot of it right now is aimed towards taking technology and just selling it to the soonest pharma company that might want to buy it so they can take it back to where their headquarters is and do what they want with it.
Yeah.
And I just think that that's a missed opportunity, because I think that there's a higher ceiling on for Australia than just being, just selling off pieces of the innovation to other groups.
Yeah, yeah, definitely. And I mean, I think one thing, as you mentioned, because there is an existing relationship between Novoron and MStranslate, we'll, throughout this journey, be able to keep having these conversations and give a bit of insight into what this is like. Because as you say, I do think it is interesting for people to hear what this process is like. It's not as simple as Travis just getting on a plane and then the research starts happening. There's going to be a number of steps, but we'll keep people up to date.
It's actually kind of terrible. Vivian, you were correct. Very, very true. And I was actually really impressed. And Hobart's a surprisingly cute town. I really liked Hobart and
in the country of Australia.
In the country of Tasmania. It was Brett makes fun of me because I was maybe a little bit confused that Tasmania was a part of Australia at one point, but that was a long time ago, and I've learned now. I've evolved.
But yes, Vivian, we have obviously, again, very early stages, so we can't talk too much about what's going on, but we were down in Hobart. We had some excellent conversations with some people down there at the Menzies. So again, whenever we're in a position to talk more about that, we will.
Yeah, we'll probably make you guys the first to know, I'm guessing.
There you go. Exclusives are going to be coming out left, right, and center through MStranslate throughout this process. Let's jump away from this for a second and answer some community questions. So as we go through, we're going to be talking about the Novoron activities here. We're going to be talking a little bit about ECTRIMS, but also we're going to be answering any questions that you have. So if you have any questions about multiple sclerosis research, just type them into the chat and we promise we will get to as many as we can. As I said, we do have a slightly limited time today, so we're not going to be able to stay on for hours, but we'll get through as many as we can. So Sarah has asked, why is it that some people go downhill straight after HSCT and it seems to make the MS angry for a while when there's no T and B cells left until they repopulate, and most do really well? Do you want to jump in with that? Do you want me to jump in with that?
I think you probably are more familiar with this, but my question would be, do we know that it's their MS that's angry? I mean, that's a pretty aggressive procedure that is just not, it's going to make all of you angry.
Yeah, and there's actually, I mean, I've actually seen some data to suggest the opposite, that there seems to be some improvement post HSCT that then normalizes back to where they were. So interestingly, I guess when we think about HSCT, we think about it basically being another form of a disease modifying therapy, where we're trying to stop new damage from occurring. It's something that we need to separate from some of the research that we're talking about Travis doing, where we're talking about myelin repair, where we're actually trying to fix some of the damage that's there. But in some of the data that's come out, you do see this little bit of a rebound, where when they look at the EDSS, so the disability scale, just after in the little bit of time just after the procedure, there does seem to be a decrease in EDSS, so the people have some sort of clinical improvement rather than just stabilization that then seems to get better. So I haven't seen too much around people getting significantly worse afterwards, but as Travis says, the feeling worse is probably a lot to do with how severe the procedure can be.
Well, and is it possible that it might be a bit of a semantic problem where it depends on what you're talking about after the procedure? Because if we're talking about the ablation portion of the procedure, you're going to be sick, right? Versus if you get through the full reimplantation component, I think that's what you're talking about, where you actually can see some improvement.
Yeah.
I I would imagine that if there's, because I agree with you. I mean, my understanding was that people do get a bit of a bump after the reimplantation of the tissue, but you're almost certainly going to be pretty sick during the rest or up until that point. So.
Yeah. So Sarah's just added they talk about it on the post HSCT groups, especially spasticity. I mean, I haven't seen anything on that. One thing I wonder is it would be interesting to look at the activity levels of people pre and post. There may be a bit of a decrease in terms of what they're able to do sort of when they're going through the treatment, and just after they may be a bit limited, and that may sort of lead to some problems with spasticity, but I haven't seen any specific. If you can forward any research or studies on that that people are talking about, we'd be happy to take a look and provide some comments. I haven't seen any.
Yeah, I haven't seen studies. Like my guess, and this is a guess, I don't take this as like expert advice. My guess is that the anecdotal experience of individual patients is not necessarily going to correlate with true disease symptomology, because I think supervising physicians are probably going to expect a certain level of normal physiological reaction to the types of procedures that they're having.
Yeah.
I mean, these are HSCT is aggressive. I mean, you're going through some tough stuff. So the fact that people are their bodies take a little bit to bounce back, and they're not necessarily at full strength immediately. Like I would imagine that things like spasticity and maybe even some increased pain or fatigue would be incredibly normal for anybody undergoing some similar forms of stem cell transplant stuff.
Yeah, and that stuff probably not directly related. Bringing it back to your initial question around the B and T cells, and if they're gone, why is this happening? There's probably other mechanisms at play there that are beyond the autoimmune response that we're expecting has disappeared through the treatment.
But that was just but that's not to discount the kind of like patient reported findings. If it's possible that we rely a lot on for HSCT specifically, because nobody does it in the States really.
Yeah.
We rely on publications, so I don't get a lot of patient interaction. So.
Yeah, and I mean, I think that sort of stuff, and we talk about this a lot, and it's something I think we will probably see at ECTRIMS again this year, is that there is growing awareness of the need to manage symptoms.
Yep.
While there is an important part of treating the disease and trying to understand the disease better and coming up with treatments that directly target that. I think more and more we've seen research funding and research direction go towards symptom management, understanding that while all of that stuff is going on, we need to try and find ways to better improve the quality of life for people living with MS.
Yeah. And I would say, I mean, if to go to the other side of the spectrum, like I would say I think that doctors might presume that it's going to be a rough experience going through HSCT. And so it's possible that there is some patient reports out there that are worth paying attention to. And again, I don't know, right? So because I've seen these where, the level of discomfort and, recovery should be something that we talk about more, but this is all so new, right?
Yep, definitely. Thank you for the question, Sarah.
Somebody asked something up above too.
Okay, let me scroll up. Luckily, Travis is paying attention.
Yes.
So that was Sarah as well. Can Lemtrada cause cytokine release or a cytokine storm? I haven't seen anything on it. Do you?
I'm currently blanking on which one Lemtrada is.
Lemtrada's Tysabri, isn't it?
Oh, it's Tysabri? Oh, Tysabri can cause all sorts of problems. I don't think... I'm
bad with my names.
If it's Tysabri, it can, your body can have all sorts of not fun reactions to it.
Sorry, Lemtrada's alemtuzumab.
Oh. Then I don't
know. I mean, alemtuzumab again it's quite an aggressive therapy. I mean, it's similar to the others in terms of mechanism. It's wiping a lot of stuff out. It's basically a chemo style drug. I haven't seen anything specifically on it. On Lemtrada and alemtuzumab.
In general, though, are we talking about a B cell depleting drug?
It's B and T cell.
B and T cell. In general, you're talking about getting rid of cells that would be largely responsible for cytokine production. So it's a little bit of a um, it doesn't make a ton of sense mechanistically that it would lead to cytokine storm. But sometimes when cells die, they spit out a bunch of cytokines. I don't think cytokine storm, though, like a true cytokine storm, is actually kind of an acute, almost like a shock-like phenotype. So I've not heard of that. And yes, Tisabri is very safe. We handle it very well, but you do have to monitor it for a lot of like people can develop kind of like adverse reactions or neutralizing antibodies that sometimes can be inflammatory. Yeah, like Tisabri's also been around forever. So anytime that you have more data on something, you're going to see more negative things that come out of it.
Yeah, I mean, I'm seeing some small reports in the literature talking about cytokine release syndrome as sort of a side effect of alemtuzumab, but it's certainly not something that's been widely published. Let us look into that, and we'll
That's
do a
also
post on it.
that's a tough thing for doctors to even pick up on, though. Cytokine panels are not super common. They're not they're not like a standard panel of things that doctors usually order, and they're snapshots. Like they can vary from 1 hour to the next. So if you're seeing reports on it, it's definitely worth. I mean, even just like case study reports,
Yeah.
it's definitely worth paying attention to because it's not something that's easy to kind of follow. But mechanistically, a lot of those cell depletion therapies, the only thing I can really think of is that you're just getting cell death cascades.
Yeah. All right, let's go back to. So this one's a great question for you, Travis.
Which one?
So Patricia's asked, how long do you think it will be before we are able to rebuild our myelin?
I mean, I think when I started doing this in like the mid 2010s, I feel like people thought it was possible. They're like optimistic that it might be possible, but nobody really had a vision of when it was going to happen, right? Like I feel like people were like, "Wow, this is a feasible thing that we're going to be able to do," and now I feel like people are like, "Any day now," honestly. Like, I mean, I think even going back to opicinumab, or wait, no, anti-lingo, is it opicinumab?
Yeah.
Was showed a lot of promise that we can do this. It's just I talk about the anti-LIGO therapy a lot, but I think it's just about finding the right fit because it's not just about finding a drug that works. It's about also figuring out the best way to use that drug to make sure that you get the most efficacy with the least side effects. And so, I mean, like I'll tell you, from Novoron standpoint, like we think we have a drug that could be efficacious, but it's difficult because one of the things I really want to center here in Australia is I want to have a dedicated unit that's going to really tease out what is the best way to use the drug, and that's not necessarily super easy because rodents aren't people, and like without those are the models we're kind of stuck with in terms of teasing out, how are we going to use this in an actual patient, and it's not always apples to apples. So like I would say for Novoron, we really hope that we might be able to get into clinical trials in 4 years, 2 to 4. Yeah, I always like saying 2, but like let's call it 3 to 5 years, just because I don't like I in my head I always think I can go faster than I can. But there's other people that are, might get lucky, and be able to just figure out the right way to use their approach sooner. But yeah, I mean, I definitely think the clinical trials that are going to be on the horizon over the next decade, all of them, I think you should be thinking these aren't flyers. These aren't Hail Marys. Is Hail Mary a thing in Australia?
Yep.
These are, any one of these might actually be the thing. So
yeah, I mean,
or at least a thing.
I think one of the biggest shifts across that last decade, as you say, is it started as a, can we do this? Do we have any hope of doing this?
Yep.
Even though we want to. Even though we haven't seen a heap of success in clinical trials since then, what we have seen is that it is possible.
Yep.
I mean, anti-lingo showed it was possible, even though that trial didn't work, and we're not going to go. Otherwise, we'll talk for the next 2 hours about that trial specifically, but at least it showed that it was possible. Similarly with the bexarotene trial out of Cambridge, even though that was a very dirty drug and there were way too many side effects, they saw in a population of people that remyelination was possible. So we're now at the stage where we know it can
And that's
happen.
a small molecule, right?
Yeah.
I mean, so that right there. I mean, you can always evolve small molecules.
Yeah. So it's now just, as Travis says, finding the right ways to do it and making sure. And Travis can comment on this, but the biggest thing, it's always this
Hey,
balancing
Charlie
act.
is here. Hey, Charlie.
It's this balancing act of we do understand as much as we can that we want to have these things available as soon as possible. We understand that the delays are frustrating and that everyone wants them to be available now. But as Travis has said, and he will add to this, what we don't want in that rush is to have failed trials where drugs that will work never get pursued because the trial design was done badly. And you've talked about that. I mean,
Yep.
that's what keeps you up at night.
I mean, and I think that's the thing that makes Novoron different. And I don't need to go on that soapbox again, but I think that the I think that it is an unappreciated source of failure in drugs in the neurospace in particular, not just MS, of you know suboptimal, not because I don't want to say like they're bad trial designs. It's just that I think sometimes people get a little bit too aggressive or a little bit too optimistic, and they don't necessarily understand the consequences of a trial failure and what that can mean for a program. And I think it's really important to be as certain and conservative with your approach. And when I say conservative, I mean do not oversample your patient population. Stratify as much as possible. Don't generalize too much what you think your dose strategy can be. Make sure that you do as many studies as it takes so that you understand the dose range that's going to be the dose, the dosing route of administration. Like, I think these are all things that need to be super well considered and sometimes aren't. So I think that it's companies that learn from those lessons are going to be more likely to succeed. And unfortunately, big pharma doesn't always seem to be the most agile in that sense, but that's okay.
Excitingly, we can also talk while we're talking about remyelination trials. Both Travis and I, post ECTRIMS, will be visiting the team at Cambridge and looking through some of the work they're doing at the moment with their metformin clemistine trial.
Super excited about that.
So we should be able to provide some updates, and we're hopeful to get some interviews with the team there, giving us some updates on how that trial is going. We know that it's a trial that everyone in the MStranslate community has been pretty excited about for a while. So we've been in touch with them. Things are moving in that space. So yeah, we're hopeful that we will be able to provide some updates live from Cambridge in the early part of November.
And don't get your hopes up. I doubt they're going to show us data, but I'm super excited about that visit because it is a little bit shocking how open they were to having us come in and giving us tours and walking us through their mindset. And like, I think I'm very excited for that. I think that is a level of kind of insight that is, I wish there was more of, and I'm really excited to see what they're doing.
Yeah, me too. So just going back on some other comments, Charlie's looking forward to a photo with us and Dr. Katz. So if anyone doesn't know, Dr. Joshua Katz is a US-based neurologist that Travis and I had the pleasure and privilege of being on a Zoom call with Charlie's group, MS group, where we sort of the three of us just went round and round and round talking about different topics in multiple sclerosis. It was the first time I'd met Dr. Katz. I think it was the first time you'd also had the chance to talk to him.
Yeah, but he's a great guy.
Great guy. It was a fantastic
to
conversation. He will also be in Amsterdam, so we're hopeful that we might be able to share a discussion with him as well while we're there.
And he is very much of the same mindset as you are in terms of he just comes from the medical side where he's very, very motivated to engage with patients and share his experiences, and the level of recall he has. I mean, he seems to be very on top of really cataloging his experiences with patients and making sure that he can kind of like reconcile his personal experience between things that are out there in the literature and then be able to spread his interpretations of those dichotomies to the outside world. And I just think that's amazing.
University of Cambridge in the UK, Charlie. Sorry, we're not going to be that close to your neck of the woods. University of Cambridge, in the UK. So there was a question here from Karen. How new should the damage be in order to remyelinate a nerve? I would imagine the older the damage, the harder it is to repair. Travis, I'm going to let you start with this.
So I have a lot of thoughts on this. So I want to start by saying the newer the damage, the obviously, like, I think that is going to be the focus until we get a good remyelination drug. Our primary focus is going to be on repairing newly formed lesions. I think that that's the lowest bar, and I think the lowest hanging fruit in terms of being able to not only be able to reverse some of the disability that might come from attacks, but it's also going to be the tissue that's most prime for repair. That doesn't mean that the more chronically dystrophic or fibrotic tissues that are from older lesions can't be treated. As many of you might know, or some of you might know from other Novoron stuff that we've done, our other lead program is in spinal cord injury, where you would be shocked at how much kind of overlap or similarity there is between spinal cord pathology and MS lesion pathology. And I will tell you that, like, in terms of the way we look at spinal cord injury, we're going for the acute injury paradigm first, mostly from a clinical trial standpoint, because we think it's our best chance to get statistical significance of efficacy in a trial population. But our long-term vision is to apply the same type of therapeutic into a chronically injured population. It's just likely that we'll need some sort of secondary intervention because the lesion largely becomes fibrotic or scarred, whatever the term is you want to use there. There's just cell types or sometimes just cavitation that takes place where there's actually no cells left anymore, which is even a bigger problem. That needs to be addressed before you can actually try to restore the tissue that's been lost. And I think that is going to be true in MS as well. The difficulty with MS is that it's easier to go and do a surgical intervention into the spinal cord than it is into some deep tissues of the brain. But I think that there's opportunities. I mean, one of the things that we kind of already know is a plausible means of not reversing, but maybe pulling back some of the fibrotic, pathology that happens in a chronic lesion. We could do through injections. We don't even necessarily need to do through like deep surgical interventions. There's enzymes like chondroitinase that we might be able to inject, which would dissolve some of the scar tissue and hopefully be able to restore kind of a permissive or at least a regenerative growth state. But in general, the problem with a chronic lesion, a chronic or a nerve that's been damaged for a while, is that you've got a couple things going on. One, it's kind of given up trying to get better. It's in the first days to weeks where the nerve is like really trying to fight for its life, and then once the fibrotic changes set in or the scarring sets in, it kind of just settles into what its new normal is. And the second is the scar, and it's very difficult to you think about it like if a bomb hits a hospital, you want to do really good kind of like demolition, not demolition. You want to clean up the mess first before you start rebuilding. And if you are in a hurry and you just dump a bunch of concrete in there, it's going to be really hard to build a new hospital over this concrete. You'd have to remove all that concrete. That concrete would be like the scar, so that you could actually do a more elegant building and get a good foundation and a good structure. So. I think in general, unfortunately, for patients who have been struggling and waiting for a long time, the first thing that we're going to, once we get a drug that actually works for remyelination, the very next thing you're going to start to see a lot of people doing is figuring out then what other things do we add to that therapy so that we can treat longer term damage, so that we can actually restore functionality or at least give some hope to reversing some of the dysfunction in more chronic lesions. Does that make sense?
Yeah, I mean, I think the key thing, and we discussed this in our last chat as well, is that there is, when we think about remyelination therapies and we think about are there going to be limitations to this, at the moment there's nothing that I've seen or read and nothing that you've seen or read or from your own experience think that is going to be necessarily a barrier. There are going to be, as you say, easier bits and more difficult bits, but when we think about things like old versus new damage, when we think about things like relapsing remitting versus progressive MS, we're not at a point here where we would say, a remyelination therapy is not going to work for someone with progressive MS. We're still very hopeful that will be the case. We think that there will be there, but just in the same way as we have other therapies, it's not going to be a one-size-fits-all approach. We're not going to have one therapy that repairs all sorts of damage for every person. We're going to need novel ways of doing it and novel ways of addressing each specific problem, but that's all going to build on each other, and once we get the first one, then we move on to the second one.
But I got to tell you, and I think everything you just said I agree with, but I think that mentality is such an obvious mentality to you and I, but I think far too often when people are thinking of, I'm going to make a drug that I'm going to take to market, it's just a drug. And they don't necessarily consider that the application of that drug might need to be variable or different or in a different combination for different things. We obviously, and the obvious, just right in our face, we see it every day. Example of this is cancer, where personalized medicine and being able to take the existing therapies that we have and be able to genotype tumors or understand different pathologies, and being able to say, okay, we know that we have these tools, but this person, based on this information, we're going to use them in this combination.
Yep.
That is something that I think in neurology in general is lacking. Mostly, and to be fair, we don't have a lot of tools yet, so it's not like that's not understandable. But I do think, to what you're saying, like, we're basically trying to create a really cool tool, and that tool is going to need to be part of a toolkit that for acute demyelinating lesions with relapsing, remitting, or even progressive, we think it will do one thing. But anything that deviates beyond that, it's going to be a different set of tools, where that's going to be hopefully an anchor of the toolkit. But it's the open-mindedness. I think that it's going to take a while before you start seeing. Neurologists are kind of historically slow to adopt creative, kind of like dynamic therapeutic mindsets, and that's not in their defense. It's because neurology is a much more complicated field where diagnostics are much more difficult. Like a cancer is a cancer cell. It's there. It's physically, you can see it. It's much more difficult to be able to see early Alzheimer's or Parkinson's or even MS. So I'm not trying to be negative about neuroscience. It's just or about neurology. It's just because there's so many kind of like difficulties or obstacles, the agility can sometimes be a little bit lacking.
Yeah. Well, and I think that it goes back to what we were saying before about trial design. If you go in with a mindset of this is going to work, this is our remyelination therapy, this is the remyelination therapy, then you're going to have a whole heap of participants in there that may not benefit from it, and that's going to affect your ability to see whether or not your drug actually works. So yeah, there's lots of things that come into play here. Again, we could talk about this for forever. Charlie,
thank you, Charlie, for your kind words. I don't think everybody agrees with you, but I'll take it.
Some people would suggest that we tend to ramble on a little bit,
or at least, especially me. That's kind of you to say it's both of us.
Well, I was a bit shocked at the start of that when you said that you agreed with me. I think that's the first time you've ever said that.
Yeah, don't expect it to happen.
Yeah, I'm not. So Nicole's asked, what's the most unexpected challenge that Novoron is facing right now, other than me?
I mean, to be honest, that coming here and actually having to go to these meetings with you, that was an unexpected challenge. I expected it to be like professional and like, No,
you didn't.
Obviously, the biggest problem for a small startup is always money and opportunity. But I would say the most unexpected problem, there's so many potential answers to that question.
It can even be in the US.
Yeah, in Australia, the challenge isn't unexpected. It's just trying to figure out. It's trying to take note of all the potential opportunities and figuring out what combination and a path is going to be the most beneficial and efficient for Novoron. But in general, like I would say, the biggest unexpected challenge for Novoron, I would say if you were to take it back beyond just the right now, but over the last year, our therapeutic target is LRP1. And to be honest with you, what made us very unique is that we thought we could target LRP1 for remyelination and spinal cord repair. But LRP1 is a lipoprotein receptor that the reason we were so unique in the space is because people thought lipoprotein receptors weren't good therapeutic targets. And there's been some findings, especially in the Alzheimer's space, that have kind of drawn more attention to LRP1. But there's an entire family of lipoprotein receptors that have been implicated in other pathologies. And we've long thought if we could successfully target LRP1, there was an opportunity to be able to target some of these other family members for other diseases as well. And we've been in the background is kind of like our deep R&D efforts, like the basic science stuff that we thought that was high risk, maybe would never work, but we thought it was worth a shot. We're trying to develop a platform where we could surgically target these receptors so that we would leave their normal, healthy biologic functions intact, but be able to disrupt disease-based activities. And we've spent the last year really focusing on trying to do that. And so now we've been able to prove that we can. And this now, so being able to pivot from this very specific, basically CNS specific drug company where we're taking a very select potential group of molecules and developing them specifically for neurologic conditions or damage. We're now looking at potentially being able to create drugs against a variety of conditions, and that growth, that expansion, that's pretty intimidating for a company that's been getting by largely on grants for the most of our existence. But we have really great investors. I would say that we have gotten extremely lucky. I think it's fair for me to tout Two Bear Capital is our first big private investor. Backstage Capital was our first investor with a very small seed amount, but Two Bear really came in with the first real funding, and we're now preparing to do a second round with them, and they have just been a tremendous support. And to be honest, they've given us a lot of the encouragement to take this leap and be able to do something much bigger. Yeah. And I really appreciate that because it ended up working, and like that's insane. But this mindset pivot from like, now how do we build this drug development company? The drug, not just actually, no, drug discovery and development company. The discovery part was the part where we didn't necessarily have as much of before. Yeah. And be able to maintain the level of focus and diligence on the programs that we currently have. But again, I think that's why Australia is so appealing to me, is because I think that with Novoron in San Diego really having this expansion opportunity and doing these cool things, I really think by taking advantage of a place where we can embed a team to focus on the specific disease of interest and make that their only focus, right? Like where they can work in collaboratively with the other sites that we have, but then drive this ball forward. I think that's really important because I've seen how having too many projects under one roof that aren't necessarily, you want to have a flat organization, you want to have people that collaborate, but when you have too many things going on, it's too easy to lose sight of the really critical next steps in specific projects. And so I think that that's really why Australia has appealed to me. You like how I brought it back to Australia? So that I can make sure specifically on the MS front that this project gets dedicated focus.
All right. We are going to be a little bit pushed for time. We're going for another 10 to 15 minutes. So get in your last questions as we go through, but just going back, Sarah, does anywhere closer than France allow private referral to 10 Tesla MRIs? Around $1,500 in France for it. I have no idea.
Honestly, that sounds like a good price in France. But
yeah, I don't. Sorry, Sarah, we can look into it, but it's not something I know about the location of 10 Teslas around. I know we have a 7 Tesla for research in Melbourne, but I'm not sure of 10.
Sorry, I wish I knew it's something better there too.
So just continue to scroll down. Menu. Relapsing and remitting for 9 years. Had Maven cladding fusions. New scarring in my spine. Advised to have some more treatment. No scarring since initial diagnosis. Worried about next treatment. Don't want to anger MS. It has made me worse.
I mean, that seems like I'll say on the without diving into any expertise, I would think it's good to point out that your anxiety is completely rational. I think the question you're asking is completely, that's the right question.
Yep. And I think in these sort of instances, I don't have a clinical background, so I'm mostly talking about the research. But these are the instances where I think the MStranslate community, and I've seen already that someone's given their comments, so leaning on the experiences of other people who have gone through similar things, and also just making sure that you have open and honest conversations with your neurologist and your healthcare professional around how you're feeling and your concerns is the best thing to do in these situations.
Yeah, I would say as a general rule, I think most MS patients are good with expressing themselves. But I think sometimes when they express themselves, frustration and anger are easier to express than anxiety and fear because it's a stoicism. I think that can be a disservice because I think letting your physician see the anxiety that certain decisions might bring to you, it's an important data point for them, at least for the good ones.
So Patricia, given the majority of people with MS are impacted by fatigue, what research is taking place that will improve fatigue for people with MS? It's something I'm interested to see at ECTRIMS this year. I think one of the biggest things is the fact that there is research looking into this now. I think for a very long time, when people living with MS would go to their neurologist or healthcare professional and say, I'm really struggling with my fatigue, the response was, well, yeah, you've got multiple sclerosis. That's just something that comes with it. And I think more and more now they're realizing that there are factors that contribute to this fatigue, and a lot of those factors can be targeted and can be managed. So we've seen research, probably not surprisingly, come out to show that sleep is a big factor that contributes to fatigue in people living with MS. People living with MS have a lower sleep quality than the general population, and the worse the sleep quality, the higher the levels of fatigue. Sleep is something that can be targeted. There are interventions there. So I think there's a lot going on in that space. A lot of the more traditional pharmaceutical approaches to fatigue have shown not to be effective.
Or just overly stimulating.
Yeah, so I think there are things that are coming out that are looking at how do we actually target the factors that are causing that fatigue. And I think we'll see more at ECTRIMS.
I'm gonna take a bold stance here that deviates quite a bit from what Brett said. I think fatigue in general has been an underappreciated aspect of a lot of disease pathologies. And I think in particular, especially when you consider that we now have this pretty strong link between post-viral infection and MS evolution. I think that there is a variety of pathological fatigue and chronic fatigue syndromes, myalgic encephalomyelitis, all sorts of these types of syndromes that I believe, are largely grounded in similar underlying pathology. And I think that if you really want to look into fatigue research that is going to actually make a dent, I do not think you're going to see it in MS tailored fatigue research. I think you're going to see it coming out of COVID. I think COVID, one of the few really amazing things that's going to come out of COVID is a new emphasis and reinvigorated interest in pathological fatigue from long haul COVID and people that are struggling for years, which we've already seen with other conditions, but on a much lower scale, so they never really got the attention they deserved. I firmly believe that the progress that we're going to make in long haul COVID research is going to have direct, apples to apples, applicability to fatigue related conditions like we see in MS. Yep. So I would highly recommend, because I know MS patients, in your audience in particular, is very motivated to follow research trends in the field. I would follow, and the NIH, the National Institute of Health, is actually put a whole new group together on this, and I follow them on Twitter, and I think that they're doing some amazing work. I would recommend, and I'm blanking off some of the specific people I follow off the top of my head, but I would highly recommend following that line of research, because I think that those mechanisms are going to have a lot of overlap.
Yeah. So George has just said basic HSV infection is a fatigue trigger, which segues nicely into our last question that we might talk about today around the ATA188 therapy, because obviously one of the biggest findings in multiple sclerosis research this year has been around the role of Epstein-Barr virus, which is the virus that causes glandular fever, which obviously, if you get glandular fever, comes.
Is that what you guys call mono?
Fatigue.
Is glandular fever?
Sorry, we call it mono for
glandular fever.
Glandular fever.
So just for the rest of you people who have never heard that term, that's mononucleosis. Yeah. Just so you know, it's the kissing disease.
Which obviously comes with quite intense fatigue through that. So there's obviously parts there, and from the early trials that have been done with not necessarily ATA188, but the other cell-based therapies against EBV that were done here in Australia by Professor Michael Pender, his team, where they looked at culturing up an individual's T cells in the lab to make them essentially hyperactive against Epstein-Barr virus, reinserted those T cells back into the individual, and found that led to improved and almost complete clearance of EBV. One of the things that they did see in that individual in terms of clinical benefits was an improvement in fatigue. So in terms of general thoughts on the ATA188, I think it's a really interesting trial. Similar trials also being conducted here in Australia. We're just waiting on results for that. I know that there were some preliminary results published by the Atara group in the US. That led to their stock price crashing because everyone took them as being really negative. My read on that wasn't that it was negative. It's just that they talked about results that really weren't ready to talk about yet. They were always going to be looking at a longer time frame. So I think we will wait and see. I think it'll still be a little bit of time before we hear more, but we might get some data updates at ECTRIMS. It'll certainly be something that we'll be looking out for to provide updates on. So with that being said, we might draw today to a close. But thank you very much, Travis, for joining us and participating. I know that our community love getting to hear from you. This is one of the first opportunities they've had to get to interact with you directly, but it certainly won't be the last. We may look to do one of these live broadcasts from Europe when we're there.
I mean, we're definitely going to do that.
Okay, Travis has decided, and he gets to.
We're going to be there for like weeks.
We'll do it.
I thought we were going to be able to do it on the train until I figured out how much the train costs.
So maybe not. We're not going to do a 3.5-hour train ride Facebook Live. I don't think anyone needs that from us. But thanks everyone for your participation today and for commenting with all your questions. It's been fantastic. Obviously exciting news about Travis and Novoron in Australia. Exciting news about what we'll be doing at Ectrums and providing you updates. But we look forward to talking to you all again soon. Thank you for joining us. And thanks everyone. We'll talk again.
Bye everybody.